Okay. So yeah, hi everyone. So I'm Bhairavan. So I'm a postdoc at GIS, and NCCS here at Singapore. So today I just give you a brief talk on mostly on single cell biology and single cell omics and trying to understand biology in a single cell resolution. So I'd really like to thank Shiva sir for giving me the opportunity to talk about this here. So yeah, I'll just probably just go ahead. So my talk is very brief, so I have not gone too technical about uh, a lot of things. So I have just tried to balance out on what you can learn from these technologies or techniques, and how do you generally analyze the data in overall, just so that you have a balance of both. And if you want to really look into a certain techniques or certain technology from what you see here, you could later read up on that and in depth of what you want to do and what you can do with that. So yeah, so cell biology and so yeah, so I'll like to one second. Okay. So yeah, so I'd like to point out to you. So this is basically a notes from Francis Crick when around 1956-57, when they were trying to decipher the central dogma of biology, which is basically DNA gives rise to RNA and RNA gives rise to protein, and it can never happen this way that is shown here. And also DNA and RNA can, I mean DNA basically replicate to make another copy of itself, and this is what the central dogma is. And once we started knowing, once we are clear with what the central dogma was, if you look at DNA, RNA, and protein, what they are just a bunch of sequence. So DNA is basically made of four nucleotides, ATGC, and RNA is just AUGC. And proteins are basically a triplet codon of your RNA, which then comes up into 20 amino acids. So roughly, if you just look at it, so basically the whole of evolution and all the living organisms are encoded by just the basis, either it's DNA, RNA, or protein. And so basically the key to understand how certain things happen biologically, or certain processes happen, or how cells divides, or any other processes that's happening within the cell to a scale of organism. So you need to technically understand the sequential arrangement of these bases and what these bases can correspond to, which is basically your RNA and messenger RNA here, which we call them as genes. And what do these genes code, and how do we get proteins out of that? So for the last 50 years or so, we have invested quite a lot of time and energy in understanding the sequential arrangement of these different RNAs and DNAs and modifications in DNAs, proteins and proteins modifications, and so on. So today the talk that I'll be talking mostly about RNA sequencing, and mostly in a single cell level. So I'll just give you a brief history of how it all started. So Sanger sequencing was the first sequencing where we started sequencing DNA. That was around 1977, and protein sequencing was again done by Sanger, but it was much earlier than this. And post Sanger sequencing, so slowly around 83, in 83 we started, PCR as a technology came up, and we started sequencing genes, we started knowing sequences from the Sanger sequencing, and ABI evolved. And the first draft of Human Genome Project started somewhere around 1990, and in between we had Solexa, which is again another sequencing platform. And first draft of Human Genome came around 2002, 2003. And around 2003 to 2005, there was a lot more advancement in technology and everything, and we started getting NGS, what basically it's next generation sequencing, where we were able to sequence a lot more bases, or a lot more lot more materials in a higher depth, which enabled us to understand of identifying genes, trying to find out what functions these gene might be, and cataloging all of them. And the very earlier stages of single cell RNA-seq started around 2009, where people used laser dissection or sorting and then sequencing to identify what RNAs that every individual cell types might possess. And the boom of the single cell technology kind of started when DropSeq was invented from or founded from Aviv Regev's lab, and which later 10x just monetized that, and then they made it into 10x genomics. And a lot of the 10x library protocols are droplet based methods, and that happened around 2016. And also the start of Human Cell Atlas, and two major landmarks in terms of understanding human uh, human cell types, organs, how do they work and function. Started with Human Genome Project, and then again with the boom in technology where we started doing single cell sequencing. Human Cell Atlas was co-founded by Aviv Regev and Sarah Teichmann as a huge consortium where they decided they'd sequence from across the globe from different human beings, different number of cell types, trying to catalog cell types within different organs, tissues, and everything to put together. And later, after 2016, we started getting spatial RNA-seqs, where basically we are trying to get single cell level RNA expression, but within a tissue where we can map within the tissue where these cells are present. So that was somewhere around 2009. I'm just giving for slide sake, but there are also other techniques. And currently, StereoSeq and Xenium are the current hot techniques in spatial RNA-seq, uh, in which people use to identify spatially in a single cell resolution how cells are arranged in a tissue and what are the cell types and how do they interact and things like that. So so yeah, today I'll be mostly talking about single cell sequencing and what are the what is the experimentation? How do you do it? How do you get single cells? How do you sequence? How do you get number of genes within a cell? And what can you do with that information? What are all the downstream biological inferences that you can make from that? And also, I'll give you a very brief overview of spatial RNA sequencing techniques that you can use to again try to map these single cells over the tissues. um, yeah, so okay, so I'll also just briefly give you okay, I'll also just wanted to give a very brief uh, intro about bulk RNA sequencing and what all. So a lot of uh, the the basic idea or the techniques that was used for bulk is just scaled into single cell sequencing, and those helped us to understand a lot of things in a single cell scale. So imagine this is a eukaryotic cell where your DNA is open and your Pol2 is transcribing an mRNA. And so this is basically start of your gene. And so if you really look at it, so your gene gets transcribed into a pre-mRNA which has a 5 prime and 3 prime end, and which has both exons and introns. So this is called pre-mRNA. So it undergoes a process called splicing, capping, splicing, which removes intron, and also the 5 prime end gets capped and 3 prime end gets polyadenylated to make the mRNAs more stable. So then this gets methylated by metal ions, and then later this gets exported into cytoplasm. So once it reaches cytoplasm, it interacts with a bunch of RNA binding proteins, and that can design the fate of your mRNA. And again, it can bind to small RNAs that can either repress them translationally or just degrade the RNA. So this is what an entire, say, a, a miniature cellular process is the main key cellular processes. So from bulk RNA-seq or from existing genomic technology, so there are multiple sequencing that can capture a different snapshot of these processes. So first is basically whole genome sequencing that just gives you uh, the ATGC arrangement of the entire genome from for all the chromosomes. And exome is basically you sequence only your mRNAs, the coding regions, and then try to find out what mutations that might have in case of, say, deceased, or say in case of a population-wide study. And ChIP-seq is basically that can give you the methylation statuses that you can see here, basically active transcription or repressed transcription, or enhancer activation and things like that. So all of this could be identified using these three sequencing techniques. And to understand RNA, you could basically understand it at multiple levels. So one is basically RNA-seq that can give you mature mRNA levels, which are polyadenylated, and long non-coding RNAs, which are also polyadenylated. And if you do RNA-seq by poly A, that is oligo-DT selection, then you can do gene and long non-coding RNA levels, and you can try to understand the splicing. And if you do RNA-seq by ribo-minus, a protocol in which you deplete, where you don't do oligo-DT selection, but you do a ribo-minus, and then you extract all the RNAs from the cell, then you can try to understand nuclear to cytoplasmic RNA level ratio and the dynamics between that. And 3' sequencing is basically can help you identify the 3' UTRs in your mRNA, and MTAL-seq is another sequencing technique that can help you identify the length of your poly A tail, because the length of the poly A tail, at least in eukaryotes, determines the transcript stability as well as the translational efficiency. So again, there are more sequencing techniques. So You can do small RNA sequencing, which can help you identify microRNAs, piRNAs, tRNA-derived small RNAs, saRNAs from your cells. And degradome is basically try to identify the degraded RNAs from your cell that can help us identify microRNA or small RNA targets. And to identify, to at least get a snapshot of the translational levels of your mRNA. Uh, okay, you could do basically polysome profiling or RiboSeq that will tell you what's the level of RNA that is getting loaded along with ribosomes, which is a measure of translation. And again, you could do, there are a lot of RIP-seq and CLIP-seq that you could do for individual RNA binding proteins or proteins of interest that would tell you basically the protein binding site in your mRNA and how that might actually influence your levels of your mRNA or the translation or the degradation status. So this is just some of the sequencing techniques that I have detailed here, but as you can see, we have multiple techniques that uses different library preparation to understand, say, from the DNA level, DNA modifications, RNA, pre-mRNA, mature RNA, and methylation modifications. Trying to identify small RNAs that regulate these RNAs, the ribosomal loadability or how much of that gets translated. So all of this could be a snapshot of this could be gotten from different of a wide array of these sequencing techniques. And so some of this is again adapted to a single cell level later, which I'll just show you down the talk that what are all the different of these could be adapted in is adapted in the single cell level. So uh, as you can see that the RNA regulation or trying to understand DNA or RNA is much complex because there is a lot of processes that are involved and there are like multiple things that you can study that would give you like a dynamic understanding of the transcription, rate of transcription, mRNA stability, translation, degradation, and other players, and so on and so forth. So this using bulk RNA-seq, we have done it over the last 20 or 20, 25 years, where we have done it across different tissues, over organs, and even in organism scales and multiple tissues. And this is a huge data repertoire that we have currently, which is basically is like no knowledge that we have. And now, okay, so what is the need? So if we had so much of these information in a bulk RNA-seq level, so what is the need? What is the need for single cell resolution? So basically, when we do all of this in a tissue level, or say an organ level, or say in case of worms or flies, an organism level, what happens is what you get is basically these are all made up of individual tissues. Say every organ is made up of different individual tissues, and this is basically cells make tissues, tissues makes organ, and a bunch of organs make organism, right? So, a single organ can be made up of multiple tissues. So when you basically do any of these bulk RNA sequencing techniques, what you do is you basically make a mashup of that for all the different individual cell types, and then try to measure the level of say RNA level or mRNA level, microRNA level, translation level. So all of that gets averaged out in this case. So one of the major uh, one of the major drawbacks, or not really a drawback, but again, the need of the R was, so when you do say a sort of treatment, or when you have a disease state, when you have given certain drugs to certain cells, so most of the cells die and some of the cells don't die. So when you basically do a bulk RNA sequencing in this case, so you're pooling all of that difference into that, and then you're just getting one expression level. But you would be really interested into the population where the ones that have not been that have become resistant to your drug, or ones that have survived your drug, right? You want to see what those cells are, what is the RNA content of those cells. So that is one of the I mean, that's one of the scenarios where you would need single cell resolution to identify novel cell type, cell state. So the need for single cell came where need for single cell resolution, RNA levels, or say any of the measurements that we want would help us identify novel cell type, cell state, or disease resistant population that would help us either tackle a disease or understand the biological processes in much more detail. So when you do this, say the same sequencing study or a genome-wide profiling in a cellular resolution, so what you do is you individually profile these cells, and you could again then cluster them later once you sequence them, because every cell type has a unique RNA levels in them. So that's what makes the individual cell type. And then you would actually find within so if a tissue, say if an organ is made up of all of these individual cell types, if you find a disease resistant population or a population of your interest, then if you individually profile them, then you would be able to see which one might actually be the one that is actually getting perturbed by whatever the perturbations that we are looking for. So that's the need for single cell resolution and trying to understand these processes in a single cell level. And yeah, I'll just give you very brief examples of what some of the populations that we identified this way. So one of this is in cystic fibrosis. Uh, mm, so we have done so much of bulk RNA sequencing earlier where we couldn't find out this particular population that you see here because it was too less number of cells that was just getting masked by or getting averaged out by other cell types that you were. But when you did single cell RNA-seq, you were able to identify the cell type, and this was very a disease specific or disease resistant cell type, where now we would find markers for this cell type that could be used to enrich for the cells or find antibody or treatment for this. And similarly in ulcerative colitis, so we found as a type of immune cell which looked like M-like macrophages, and that was specifically identified by single cell RNA-seq, and it wasn't identified earlier by bulk RNA-seq that was very disease-specific, um, a disease-specific cell type or cell state. And again, so now we have multiple consortium that does this, where we have a HubMap consortium, there is HCA, there is where we have a consortium for every individual different organs that you see here, where we are trying to sequence different organs in single cell resolution, in both single cell and spatial resolution, and trying to identify all the individual cell types, what do they interact with, and how that could be used for understanding, say, disease biology or generally different biological processes. And yeah, so bulk RNA-seq is mostly like if you're drinking like a smoothie where you had multiple fruits that you just made into a smoothie and then you're just drinking it. And single cell RNA-seq is basically trying to dissect out what are the individual fruits that went into part of the smoothie. And spatial RNA-seq is basically how spatially these fruits are arranged. So this is basically a very simplistic explanation on why from bulk to single cell to spatial, the need for it, because yeah so it's not just trying to understand these RNA measurements, say RNA levels uh, or chromosome accessibility levels in an individual single cell state, but also like where are these cells which we find to be, say, very interesting or disease specific oriented, where are they present in the tissue. So that's also is very important, and that's where the spatial analysis comes comes handy. So yeah, so I will just give you a very brief, again, a very brief intro on what are the different techniques that was used to do single cell profiling so far. So very earlier you could do limiting dilations where you take cells and you keep diluting them into, say, over different wells, and you could, at the end of certain dilution, you would come down to single cell resolution. Another way is just basically micro manipulation, where you have a microscope and you have a pipette that's controlled, and you look the cells through the microscope, and then you start picking cells through these capillary, which could later be sequenced or say qPCR or PCR or whatever was the downstream application. And yeah, so as I mentioned this before, PAX is another technique where you have certain antibodies that can bind to certain cell types, where you use that and you sort them, and then you can segregate the different cell types and you could sequence them in bulk, where now you have a similar cell type that could be sequenced. And another method is basically microdissection, where again, under a microscope from a tissue, where you try to look at the tissue and try to pick up certain regions from the tissue, which could be later used for uh, dissociation and sequencing later. So the one that made this technique very usable or very I mean, for a lot of data sets are very broadly applicable was microfluidics, where you have a microfluidic chamber which has a bead, and it's an oil-based emulsion system where cells comes from one of the microfluidic chamber, and it joins with the bead, and it just gets engulfed into this oil droplet, which we call it as gems, which is then later, so the beads have, okay, so So if you look at any given droplet, what you would see is you would see a cell and you will have a bead. And what the bead has is basically different barcodes or oligos that are present. So what the oligo is basically, it has a unique molecular identifier which is specific for a gene, and you have a barcode for every cell, and you also have a PCR handle which is later used to do the library prep. So what happens is all of these droplets are captured, and then you do a droplet-based RT, and you get a lot of RNA that gets that gets bound to these droplets that you can see here by RT, which is later. So now your RNA will have a cell barcode, a unique UMI, and then it will have a PCR handle, which would later be used to make the library preparation. So just to give you an example from a tissue, say if you from a colon, you just isolate a colon cancer tissue from which you dissociate them into single cells, and then you again send them through this microfluidic chamber where you make these gems. And so basically, this is how a typical gem making process looks. So your cell comes from one side, and the bead that you can see here just gets engulfed into different droplets. And if you just look very carefully, you will see a droplet with a bead, and there is a cell inside, and it happens in this rapid, the, ra- ra- I mean, controlled way, so that ideally you get a single cell with a single bead inside a droplet. So what you then basically do is that you take all the gems, and then so since your cells that you're isolated undergoes a lot of processes like physical and biochemical stress, what happens is the cells can undergo dissociation induced stress, apoptosis, breakage, because of which inside every gem you can have like multiple different scenarios. So I'm just listing some of them here. So an ideal case scenario where you just have a droplet with a cell inside. Sometimes you can have two cells captured inside a droplets, which we call it as doublets. And again, you can have debris, which is basically the broken cells are just present in the milieu that you have, which just instead of cells in within with a bead, what gets engulfed is all of these debris or these ambient RNAs. Or you could have cells which also have a lot of debris and ambient RNA in them, which you technically wouldn't want them to use for downstream analysis. So the idea is that you would you'd sequence all of these droplets and then later use bioinformatics methods and use QC pipelines to enrich for only the ideal cell types, which would be later used for downstream analysis. So once you get these gems, right, what you do is that you make an RT reaction inside for every gem, so that for every cell type, RNA is just get reverse transcribed and gets attached to the uh, bead and the UMI that is there, and you break all the emulsions, and then you do cDNA amplification for all the cells that you have done, and then you do library preparation and sequencing like how you do for bulk RNA-seq. So typically in a single cell data, you would have three FASTQ files instead of two, like what you see from bulk. You will have an R1 and R2, which is paired read R1 and R2, and you'll also have an index file which would tell you the cellular index that was part of your library prep, the barcoded ones that you had to identify cell specific um, to identify RNAs that belong to a certain cell. So technically, if you use a 10x protocol, you use Cell Ranger, which is a program that is part of 10x pipeline that would help you to take these FASTA file, align them back to the genome, and then try to give you cell type wise, uh, uh, cell wise individual RNA counts for all the RNAs that you have used. So again, in Cell Ranger, so how it works is that so this is basically a barcode rank plot. So every barcode is an individual cell that you have sequenced or individual droplet that you have sequenced, and this is basically a number of reads that map to that particular droplet. So it uses this barcode rank plots to actually identify which are all the actual cells and which are all basically the background noise that I had initially shown you as just ambient RNA or debris. So Cell Ranger does this to identify cell calling, individual cell calling, and then what it gives you is that so it gives you like a gene cell matrix, so where your rows are all the individual genes, and every column is basically an individual cells, and as you can see, the expression values are actually given here, like number of reads that map to that gene. So in another summary, if you just look at it for every cell, it will tell you three major things. One is basically N count RNA, or NUMI is what it's technically called. It's basically the number of reads that map to that belong to the cells, and N feature RNA is basically number of genes that you can identify from the cell, and percentage mito is basically number of percentage of reads that are mapping to the mitochondrial genes. So why this is important? Because the QC steps that we do for single cell analysis is mostly done based on the number of genes that you have per droplet to decide whether this is an ideal cell or not, and also the percentage of reads that are mapping to mitochondrial RNA. So the higher the percentage of mitochondrial reads, which means that the cell is more stressed or the cell is less viable. So we would basically discard higher mito percentage reads and try to retain lower mito percentage reads in general, but that's not true for all the cell types or metabolically active cells and so on. So once you have made the library preparation and have gotten to this level, next what you would want to do is now you want to identify for every cell what is basically the biological identity and what we can infer from that, right? So there are two major pipelines or that are available to do single cell analysis. One is called Suret, which is basically an R-based one, and ScanPy is an adaptation of Suret, but it's a Python-based one. So both of them is what most widely used to do the single cell analysis. And so basically, I told you the first step of any high throughput experiment is QC to enrich for high quality cells. So technically, most of the programs, what they look at is, as I told you in the previous slide, is basically the number of genes um, that every sample, basically that every cell has, and also the percentage of mitochondrial reads. This is just plotted over for every sample here. And they basically decide based on, for a given sample, for all the cells that are sequenced, they decide a cutoff basically based on median or mode or mean or whatever is the number. So they decide a cutoff to retain those cells or discard those cells. So the most widely used cutoff is somewhere around individual cell should have at least 200 genes. So to just call it a cell, and the percentage of mitochondrial level should be less than 30%. So also there are individual tools to do different QC pipelines, which is MIQC, DDQC, SETK, and so on. So there are multiple other tools as well. So I'll also show you another way to do QC is basically where you can make use a two-dimensional plot, scatter plot. Basically on x-axis is basically in the number of genes, and y-axis is basically percentage mitochondrial reads, and you can see the density of cells that are distributed here, and based on the mode from here, you could decide what could be the cutoff that you use for filtering your cells. So plotting the QC in this way is quite advantageous, because if you have a low quality sample or a data set, you would also start finding the increase in density of cells, which has lesser number of reads and more mitochondrial percentage, which we kind of know it's low quality cells, and you can instantly figure out the sample's quality by just looking at the QC plot. So again, I'd just like to show you. So this is a small methodology that I developed and which will be out in bioRxiv very soon. Is basically a cell type specific QC, where we do two rounds of QC. The initial round of QC is basically just trimming the extremes based on just using NODG very less and a very lenient my percent mito content to just remove the empty droplets, and then we take all the cells that pass this round, and then we identify broad cell types and we do a cell type specific QC. And we have again shown that doing cell type specific QC. The reason behind this is that every individual cell type has a unique NODG and some the number of genes and percent mito distribution. So using one cutoffs for all cell types within a tissue just might end up you might end up discarding certain tissues which have lower NODG, a lower number of genes, or also you might also end up keeping a lot of bad quality cells for that particular cell type. So if you look at these are the 7 different cell types, say from colon cancer, where you would see um, every cell type has a unique number of genes and percent mito profile. So once you have these QC filtered cells, so you would want to figure out what the biological identity of these cells and what can you learn from them, right? So once you have the QC filtered cells, the first step that you do is basically you normalize the data like any other high throughput experiment. So in the scale, you do a log normalization and you scale it to a factor of 10,000. If you have in bulk RNA-seq, we usually do it in millions, but in single cell RNA-seq, the depth is much lesser for every individual cells. So we do it in a factor of 10,000. So so once you normalize the data, so what you want to do is that, so similar to any of the high throughput experiments, so the assumption is that if you have a data and if you have a high throughput or high dimensional data, so the variation in the data is contributed by very few individual entities within the data set, and not everything in the data set would change, which basically means that if you have a cell, the if you have two cells and you're comparing two cells, the variation between the two cells could be very few genes between them, and not all the genes change to same to become cell A to become cell B. So you find highly variable genes within your data set. So usually top 2000 or 3000 is used, and so the genes that you predict here as highly variable would be the genes that you do all the downstream analysis, because these genes contribute to most of the variance in your data. So all the downstream processing would be done for these 2000 genes and not for all the genes in your data set. So once you have these genes, what you do is you scale the data, which is basically is like mean normalization or z-score, and you do PCA to do a first round of dimensionality reduction, in which you can see a PC plot where you again reduce the dimensions from 2000 to 30 or top 50 PCs that can explain all the variance in your data. So once you have this PCA, what you do is that next you try to find k nearest neighbors for all of these cells based on the PC coordinate based on the PCA, and you use an Liouville algorithm if you're using Suret to find clusters based on these neighbors. So basically, these are the k nearest neighbors, and using clustering, you would try to find cells which are very close within themselves and very different from other cell types that you see. So once you have that, so you would have the different clusters, cells are grouped into different clusters. So ideally you would think all the different clusters would correspond to different cell types that you would have. So you are representing this data again in a manifold method, which is basically UMAP, which preserves both the global variance and the local structural variance, sorry, the local variance within the cell, and also preserves the global variance of the data, where you can find how every individual clusters or cell types are separated. So once you have all of these clusters, so there are also other manifold methods which people can use to visualize the data. So the most widely used is basically the UMAP. It's UMAP currently. So once you have different clusters, what you would do is you want to know what are these clusters are, because these clusters are just numbers currently. So what you do is that you do try to find top genes that are present in all of these clusters. So how do you do that is, say if you're taking this cluster, you take the genes within this cluster, and then do D between all of the other cells that you have in the data to find what are the genes that are enriched within this blue cluster. So similarly, you repeat it for all the other clusters that you have in your data, and the top genes that you have would basically be the identity of your data set. So for, say, this is just 8 clusters that you have here, and these are the top genes for every clusters. So based on the genes that you have here, you decide what cluster 0 or cluster 1 would be, which could, again, there are multiple ways to do this. One of the ways is to just look at the top genes and what are the do they correspond to any known cell type marker that you know from earlier, and you can correspond cluster 0 to be, say, epithelial, and cluster 1 to be T and K, or cluster 3 to be myeloid, and so on and so forth. So uh, so yeah, so this is basically the first round of analysis that you do for every single cell RNA-seq data. So once you isolate them into single cells, you sequence them. Once you cluster them into like broad cell types that you have done here, what you want to do is that you also you have the resolution to understand the subtypes within every cell type. Say if this is basically all the major cell types that you have from your data set, and you are interested in, say, fibroblast. So what you can do is you can you can take only the fibroblast cells out, and then you can repeat the same analysis that you've done before, where for the fibroblast cells, you can now look for highly variable genes and again recluster the data to find individual subtypes of fibroblast within your data. So if you just do that, again, I'm just showing you broad classifications of cancer associated fibroblast from the data. So if you just look at here, just that one cluster, you grouped all the fibroblasts into one cluster, but if you subcluster them by redoing the entire analysis only for the fibroblast cells, then you have multiple individual subtypes within the fibroblast, which you can again use to understand the biology downstream, right? So yeah, so this goes on for what question you are trying to ask, what data set you have. You design the downstream analysis from this slightly differently. So from now on, I'll just give you a broad array of analysis that you could do with these clusters or these cell types, and what can you infer from them, and what biology can you learn from them. So again, just give you a very uh, brief on the work that I have done before, that I have done from the lab here earlier, is basically we work on colorectal cancer, and we work on, so colorectal cancer can be molecularly classified into two, which is basically MSS and MSI. Basically, MSS doesn't respond to immune therapy. So we are interested to look at patients that are MSS and what kind of cell types do they have, and do they have anything in particular that makes them doesn't respond to immunotherapy and things like that. So we did a huge single cell RNA-seq study where we isolated single cells, around 3 million cells from around 63 patients. And when we looked at it, looked at the tumor cells, which is basically epithelial cells, we identified two molecular subtypes from the data, which is basically from the MSS, we could classify them into ICMS2 and ICMS3, where we found a subpopulation of MSS that we call them as ICMS3 could be more closer to the MSI, which is basically the therapeutically treatable population. So, and then you can see them to be separated here quite well in the UMAP space, saying that they are molecularly quite distinct. And okay, so so this is one of the observations that we made just based on the tumor cells that we had from the colon cancer, and what we can, so molecularly do we find like distinct cell types, I mean distinct cell state within them. So there are other interesting things that you can technically try to observe from any given single cell RNA-seq data, or say any high throughput data sets. Is one is to identify the rare cell type that I had shown you before in CFTR, where you just look at, you'll be able to identify rare cell types that you can find that you were not able to find from bulk RNA-seq data sets before. And another thing is you can now track cellular processes in a very gene expression space, because uh, it, uh, in biology, everything is a continuum. So take a fertilized egg that just grows into a complete organism. It then goes through a continuous process of differentiation and self-renewal of stem cells that then gives rise to different lineages and different organs and systems. So now what you can basically do is that you can now track these biological processes in a single cell level to identify any novel populations that could be associated with any of the developmental disorders, or generally just to understand how what are the transcriptional variants that you can find in any of these processes. So here I have shown you development, or say during early stages of development, but this again holds true for the stem cell biology field, where you can have a stem cell and then differentiate into different lineages, and you can track these cells which are going through different lineages and see what kind of gene expression changes happens in a single cell level. So again, since it has a wide range of application, we have multiple atlases apart from the human atlases. There is an atlas for C. elegans, mouse, fly, where people have extensively sequenced a lot of tissues, lot of cells over, and identified a lot of cell types across different organisms. So now this acts as a huge data set for us to understand the biologically conserved process. Do we see similar cell types that we see from, say, mouse development to human development, or fly to human? And uh, the applicability is just way beyond from here. So another thing that you can do is that, so as I'd earlier hinted about the disease specific cell states, right? So assume you have a lung cancer patient and you treat them with a drug which basically say it's an EGFR inhibitor. So if these are the cells and you've treated them with inhibitor, so what happens is post treatment, so some of them would respond to drug and then they become drug sensitive cells, and some of them might be just resistant to drugs, and some of them might just be the microenvironment cells that either enable the resistance or enable the sensitivity. So now, since you can sequence them in a single cell resolution here, so you would be able to identify all of these populations individually if you do sequences. And say if you find these resistant populations, you can see what are the top genes that are present in these cells, and that could help us identify new biomarkers for a lot of disease, or biomarkers for identification of the early detection of disease. And also, if you can find specific proteins and antibodies that you can isolate these cells, then we can understand biologically how they are different from the cells that are responsive to certain drugs, and then try to identify, try to improve the standard of care treatments for a lot of things. And also another very straightforward thing that you can understand from any single cell study is the proportionality, right? So if you have a tissue and you have, say, four different cell types, and if you have treated the cells with, say, certain inhibitors or certain uh, or any perturbations, you want to see if your cell has four different cell types, how does individually these cell types change on treatment or perturbations. So that can help us understand dynamically what happens during a process, and then what cell types changes, what cell type changes can have a biological implication. And so these are again not all extensive things that you can do from single cell RNA-seq data, but some of the very interesting things that you can observe from a single cell RNA-seq data once you have done the sequencing, what all the new biology or new mechanisms that you can try to understand. So uh, okay, another challenge here is that since you're sequencing at single cell, so from a single patient you take multiple tissue blocks and you isolate single cell from all the blocks and then you sequence everything. Say you get around 10,000 cells per patient. So with just one patient, this is not going to go. So you would want to do it from multiple patients or say from multiple samples. So when you have multiple of these data sets, then you just sequence them in different lanes or you sequence them in different batches. So here comes the usual high throughput, a large scale high throughput experiment confounding factors, which are basically the experimental and the batch variations that you can generally see. So once you have, so what happens is if you have sequenced, say, a same patient's, sorry, for a same disease, you have sequenced multiple patients in different batches. So every sequencing batch is going to have its own sequencing technical artifacts. So so here, when you do the downstream analysis for, say, different experiments here, what you would see is that you would see the cells would be separated based on the batches that you sequence. So ideally, what you would want to do is that, say, if I'm working on colon cancer and I've done sequencing for over 60 patients, I would want to use all the cells from 60 patients and then see what common biological processes emerge from these patients. And because that's important for you to decide downstream therapy or downstream targeting or biomarker identification, because there is also patient heterogeneity. I'm sure some of you or a lot of you would have heard about personalized medicine and things like that, where there's going to be an interpatient variability, but also there is some molecular processes that are conserved across different patients for the same disease. So you would want to see what are all the collective mechanisms that are there. So in that case, you want to integrate the data set. So there are multiple integration techniques. So one is Suret that uses CCA, which is basically like explained here. So it tries to find anchor between these two different batches. So you have to say your cells and what are the batches, and then it tries to find nearest neighbors across batches, and then it tries to find anchors. And based on the anchors, it would just try to integrate all the different same cell types together across different batches. So as you can see, before batch corrections, the cells are separated, even though the similar cell type, the cells are separated based on batches. But once you integrate them, then there is good intermixing of cells between them. So another program that does a very efficient way of batch correction is basically Harmony, and this is quite efficient because this doesn't actually interrupt with your actual data set. It only works on the PCA space. So based on the PCS that you have, and you give the PCs that you have and the batch, and you can give for every cluster or whatever the batch is, it tries to find a centroid for say different clusters and for different batches, and it tries to correct the clusters to move the centroids closer to each other so that you can integrate them in a very efficient way. So Harmony is one of the most efficient program that is there to do batch correction. So yeah, so now so far what I've just shown you is that how do you do a single cell experiment. Once you get the data, how do you analyze it, and if you have done it across batches and experiments, how can you integrate the data. So now I'll also show you some of the programs that can help you learn some of the actual biological processes that you can look at from the data. So one of the major things that people look at is basically gene regulatory networks. So this is kind of known from bulk RNA-seq and from other knockdown knockout experiments, that a transcription factor can control a bunch of genes within a cell type, and a bunch of transcription factors can push a cell from state A to state B. So if you remember if you remember pluripotent stem cells, the Yamanaka factors is basically a bunch of transcription factors expressing a bunch of genes that makes the cell to be differentiated cell to go back to the stem cell state. So people has been a wide interest to understand these gene regulatory networks. So now, since we have an expression level in a single cell space, there is this program called CNIC that tries to identify gene regulatory networks from your single cell data. So what it does is that it takes your gene cell matrix and then try to find co-expressing genes, basically genes that have similar expression across different cells, and then it tries to group them into different groups, and then it then tries to form regulons, and it uses the existing transcription factor database from human and what is the binding site, and then it then tries to build this gene regulatory network for every transcription factors for every different groups that it has, and to see whichever it corresponds to. So once you have that correspondence, so it can tell you for different clusters what are all the bunch of transcription factors that are present in this group and what is the binding motif. So this helps us understand the gene regulatory network from a single cell data. This is this is quite useful in case if you have like a disease state, or if you have a treatment before and after treatment cases. When you see a particular cell type or a particular cell state evolving, is there any transcription factor that could be attributed to this particular cell state or cell site changes? Because if you want to efficiently design a drug, you would want to target something that would have an effect on a lot of downstream gene, which is going to be a transcription factor. So in that case, inferring gene regulatory network from single cell RNA-seq data is quite interesting, and CNIC can do that. And so this is one of the downstream analysis and biology that you can learn from single cell data. And the next one is trying to understand the cellular trajectory. So I'll come back. I'll just restate my earlier example where a stem cell is just becoming, say, a muscle cell or a neuron or a fat making cell or a liver cell. So the starting point is all the same, where it's a stem cell. It just goes through a different transcriptional changes where it just becomes, say, cell A or cell B. So now we have single. So say if you have sequenced stem cell culture over differentiation at different time points, say you have that single cell data. So you have individual cells RNA level from that. So since you have an RNA level from an individual cell type, in an individual cell resolution, you also have reads that are mapping to spliced as well as unspliced mRNAs. If you remember my earlier slide, there is pre-mRNA and mRNA, right? So basically, RNA velocity is a program which kind of uses this spliced mRNA and the number of reads that are mapping to spliced mRNA and unspliced mRNA, and tries to identify, tries to order the cell. So basically, the idea is that if a cell A is going to be a cell B, then you would have more of the transcripts that are specific to cell type B in cell type A in unspliced mRNA. So based on that, it tries to build a trajectory, and then it tries to order your cell types or your clusters to say that where a cell might be traveling. So trajectory analysis is again one of the interesting analysis that you can do from single cell data, where you can see progression of a certain cell type moving into a certain cell type going into, say, a stem cell is becoming a muscle cell or a liver cell. So what happens and what kind of intermediate states that you can find based on the RNA expression data. So there is another program called Monocle, which again does cellular trajectories. So, but it does it slightly differently. It calculates something called pseudotime. So what it does is that it takes a bunch of cell types. You can take a bunch of cell types that you have, and then you can say whichever cell types that you are interested to look for trajectory, and it does a DA analysis between them, uses the DA analysis genes, and then based on these gene activity, it tries to do do graph-based methods to again form a trajectory in a pseudotime level, to again to say from cell state A how a cell goes from to state A or state B based on the pseudotime level. So again, these are some of the interesting things that you can again look from single cell RNA-seq data. So, okay, so now you have individual cell types from a tissue, right? So we know that all the individual cell types, they do talk with each other, and they are in direct contact with each other, and then they are interregulatory, or the cells around a particular cell can have an influence on the transcription or the RNA levels that are present in another cell. So there is this program called Cell Phone DB, which tries to which at least helps us to understand cellular crosstalks from the single cell RNA-seq data. Say, assume you have input RNA-seq data with three different cell types. What it does is that it has a repertoire of known ligands and receptors from protein interaction data and a lot of already known data sources. So what it does is that it takes all the ligands and all the receptors, and then it try to look for the expression levels of the ligand and receptors between these cell types. So to have a null hypothesis, what it does is that it randomly shuffles different cell types and then tries to see what is the association of a particular ligand with the receptor, and that would be the null distribution. So if actually two cell types are talking, or if there is a cellular communication between two cell types, then you would have corresponding cell type and the receptor to be expressed in a similar level between the two cell types, or a subpopulation within a cell type. So that is what you find here. And then what you do is, for every cell type pair and for every ligand and receptor pair, it will try to calculate a meaningful interaction, and then it ranks them to see, to at least tell you which cell types interact with each other and what is the receptor and the ligand interaction pair. So this is quite helpful in case if you're trying to understand immune cell types, or how immune and the tumor cell crosstalk happens, or say any two different normal cells, how a fibroblast interacts with an immune cell, or how a fibroblast interacts with the tumor cells. So with the known ligand and receptor information, we could use CellPhoneDB to infer all of these information from single cell RNA-seq data sets. So, um, so this is just single cell RNA-seq data sets. What you can learn and infer from that. So there are other single cell multi-omics things that you can learn from a single cell level. So one of that is basically the ATAC-seq. ATAC-seq is basically it's also there in bulk. You could do a bulk ATAC-seq as well. So ATAC-seq, the principle is that it tries to identify the chromosomal accessibility region that is done based on the TN5. TN5 is basically a transposase enzyme activity. Based on that, it tries to identify uh, uh, chromosomal accessible regions within your DNA. So why is that important? Because if a region is chromosomal accessible, then that region is most likely transcriptionally active, and when it is not, it's most likely transcriptionally repressed. So since you already have an RNA data from single cell RNA-seq, now if you do ATAC-seq, what you would basically do is that you can see the epigenome to RNA level correlations and try to find more correlative biological processes or genes that you can do for disease or biomarker discovery, or identifying of certain mechanisms within cell types. So this is how technically an ATAC-seq is done. So ATAC-seq analysis is almost similar to single cell analysis, but the difference comes where you, instead of quantifying individual gene level expression in ATAC-seq, what you technically do is that you call peaks for all of your cells, and you find for all the peaks that you have, you then find what is the nearest gene that is there for you to associate it to, and based on the peaks, you again do downstream dimensionality reduction and then cluster the data based on that, and then for okay, so based on that, you cluster the cells and then you could use it. So also there are, you could integrate, or you could add your ATAC-seq data to RNA in a Surat object, and you could analyze both the data set together to again to look at individual for an every cell what epigenome looks like, or the chromosomal accessible region look like, and what is the RNA profile for certain genes of your interest that you can look like, that you want to look for. So single cell ATAC-seq has so you could integrate these two techniques to again look at the biological processes and understand epigenomes in a single cell level. And so again, there is another type of sequencing, single cell sequencing. It's called site sequencing. It's basically cellular indexing of transcriptome and epitope. Here you sequence an single cell in an RNA level, like I've shown you initially. There is an additional layer where you actually have antibodies that are tagged with DNA barcodes. So basically, before you subject your cells to single cell, the droplet based method, what you do is that you put this antibody to the cell so that whichever the proteins of interest is basically bound to it, and then you subject them to do this, where you will also have this. And you could also get a pseudo. I mean, you could also get a protein measurement along with unbiased RNA levels from that cell. So now you're also profiling single cell proteins along with an RNA, and people also used to use antibodies for proteins of their interest, depending upon the project and the study that they're interested in. So again, as I've told you, this is like individual multi-omics, singles. These are individual single cell studies where you can find protein measurements along with RNA, or you could do attack and RNA and then integrate them together. So now there are multiple multi-omic studies where you, from the same cell, you could isolate, have separate barcodes for DNA and RNA, and you can separate them in a very distinct way, and then you can sequence both together and then try to understand genomic level, the genome state as well as the RNA state from the same single cell. So this is where the field is currently heading to. And as I've told you, from just the single cell biology, what we have tried or understand is that uh, uh, from a tissue we are able to break them into individual cell types, and within that cell type information we are able to infer gene regulatory networks. We are trying to we can identify cellular crosstalk. We can also look at the epigenome. We can look at uh, individual protein levels using site-seq. There is something called perturb-seq, and there are a lot more variations of single-seq that is out there, which I'm not talking because of the constraint of time. But you could do all of these measurement in single cell level. So again, the next question comes is that, so now say if we found a population that is very specifically that is disease specific or something, we want to see where it is present in the tissue, and that's where the spatial angle comes in, as I've told you earlier. So there are different two types of broad spatial techniques. One is sequencing based and another one is probe based or antibody based. So you can use that, and what you can try to understand now is that you can try to understand cellular neighborhood and how a cell of your interest, what is it interacting to, what are the neighboring cells of that, and how that influences the RNA levels in this. So very briefly, I'll again talk to you about three different spatial techniques. I mean, there are still a lot more techniques, but again, due to constraint of time, I'm just going to talk about these three techniques. So one is basically probe based, which is you have a list of genes of your interest. So you design a probe for all of these genes, and then and then what you do is basically an iterative fish. Fish is basically fluorescent immuno hybridization, fluorescent in situ hybridization, where you do an iterative fish for all the genes of your interest, and then you again stain for nucleus, cytoplasm, and membrane to identify the cellular boundaries. And then based on this fish image, these iterative fish, you just make them into quantitative data to find out for individual probes what are the number of spots that you can find within a cell. So another method is basically sequencing based, in which you have spots or regions in which you again have these oligos that you saw for single cell droplets that are similar to that is present there, and you mount a tissue on that. It could be either a fresh frozen or a PFA, where you mount the tissue on that, and then you release the RNA from the tissue to the spot where it binds to the probes that are that are there. And that again also the probe gives you the XY coordinate to tell you in a tissue space where does your the cell or the cell is present. So the first one that I talked to you about is Visium. So Visium is basically, again, as I told you, this is a sequencing based method. It is again, it has a lot of probes. It has basically spots, and every spot is basically like an oligo that is there, and the oligo has poly DT, again a UMI and a sequencing adapter and a spatial barcode. So what you do is that in this slide you would basically mount a tissue, and then you do the processes where you would release the RNA from the tissue to these spots where it binds to the spots, and then later you can sequence all the spots. And since you know the XY coordinates for the spot, you know where in tissue it is present. So for every spot is around can correspond to 1 to 4 cells. So Visium is there is Visium 2 now, which again is much more efficient than Visium, and there is also 10x Xenium, which is even more efficient than Visium to find at least 5,000 genes on a iterative fish-based method. So in Xenium, what you do is that, say if you have a tissue, say in this case a brain postmortem tissue, which you placed on that. So once you say it has white matter and it is from level 1 to 6. So if you place them on a spot and you sequence it in a single cell level, and if you look at the RNA expression level, then you are able to recapitulate the similar, the coherence between that. So Banksy is a program that does spatial analysis in here again. So I'll also slightly introduce you about SlideSeq. This is again a probe-based sequencing technique in which you have different spots that are mounted on a glass slide. Again, you mount a tissue on that, and then you release the RNA to this, and then every spot will have a UMI and poly-DT where your RNA binds to that, and that could be sequenced to give you a single cell level RNA expression level, and this gives you an XYZ coordinate. So if you just look at the hippocampus and slice of an hippocampus, you can see clear structural anatomical features and the gene expression level coherence that you can see here for say three different cell types here. So this could be used to just understand the normal biology of healthy cells or healthy tissues, and also in case of disease, how this might change or what happens. So the third one is basically StereoSeq, which is from BGI. Again, this is a sequencing based method, and this is quite efficient method. So they use DNA nanoballs. DNA nanoballs, again, instead of a spot here, the nanoballs will have is arranged or printed on a pattern chip, and every DNB is basically 100, sorry, would have around it has around 400 spots, which is around 100 micrometers, which where you can place in a bigger chip. So you have the coordinates of all the chips DNB that you have using the solid sequencing, and all of the DNBs have the probes that you can see here, where you can mount the tissue on that and you could sequence. So StereoSeq is basically currently the most high-res spatial resolution that you can find, where you get more number of reads per spot, and you can have around uh, say a lot of DNBs within a cell, which means that you can actually get within cellular resolution level of data. And also one of the other biggest advantages that StereoSeq can mount. So this is basically mouse developmental time point data that is mounted on it. So it's basically like a bigger tissue slice that can be found, and you can find all the individual cell types from DNB with a very high number of genes compared to any of the known spatial techniques. You find more number of genes or genes per cell compared to any other techniques. So I think so far what I have tried to tell you is that how single cell RNA-seq is a very powerful tool to understand like multiple biological processes, understand cell types, cell states, biomarkers, mechanisms, gene regulatory networks, crosstalks, and a lot more things that you can identify, and how spatial RNA-seq or spatial sequencing or spatial techniques can help you understand how in a single cell resolution how does this happen in a tissue level, and what are the neighboring cells, what are the interacting cells, and so on. And with a lot of data that is being generated by both single cell and spatial data, there is a very huge scope for AI. And currently, at least the field is using a lot of AI and ML methods to use these data sets to train them on these data sets to identify cell type markers, cell type predictions, doing better downstream analysis and things like that. And there are foundation models for single cell biology specifically, that is SCGPT, SCBERT, and so on, that can learn a lot from these data sets, and then that could help us fine tune a lot of specific tasks in the downstream. So yeah, so understanding biology in single cell resolution uh, does help us to identify a novel cell type, cell state that could be disease specific, or where you could identify therapeutic targets or understand new biology or how a certain process happens in a single cell resolution. So I think with this,